Good afternoon, everyone. My name is Shoshana Hollander, and I'm the Director of Data Science at Genome BC. Thank you all for joining us for the last session of our Genomics Forum called All In, Using Large Population-Based Genomic Studies to Inform Patient Care. In 2021, Genome BC hosted Dame Sue Hill, who discussed UK's efforts in generating large population-based genomics data. This year, we brought together experts from Australia and the United States of America to discuss the success stories and lessons learned regarding their large-scale cohorts. In addition, we would like to explore how Canada can leverage already existing large biobanks to have its own large-scale population-based genomics initiative, one that can support the development of disease screening, diagnostic, prevention, and treatment, as well as inform public health policies for our diverse population. We're going to start with some opening thoughts on this topic from Drs. Philip Awadallah and Parveen Bari. Then we will be joined by Tiffany Botwood and Anastasia Wise for a more detailed discussion, followed by an interactive Q&A session with you, the audience. As in the previous sessions, you can ask your questions anytime throughout the session using the Q&A panel on the side of your screen. You can also upvote questions as they appear, and we'll draw upon those most popular questions during the Q&A. I would now like to introduce Drs. Philip Awadallah and Parveen Bari. Philip is the National Scientific Director for the Canadian Partnership for Tomorrow's Health, also known as CanPath. He also serves as Director of Computational Biology and as the Executive Scientific Director of Ontario Health Study at the Ontario Institute for Cancer Research and is a Professor of Population and Medical Genomics at the University of Toronto. Parveen is the Scientific Director for the BC Generations Project and a Distinguished Scientist in Cancer Epidemiology and Prevention at the BC Cancer Research Institute. And with that, I'll hand it over to our first speaker, Dr. Philip Awadallah.
Thanks very much for that introduction and having me here, Dr. Susanna Hollander, and the folks at Genome BC for inviting me. So what I thought I would do for this presentation today was give you is largely a presentation of three parts. The first part is to present to you what we've built at CanPath, the Canadian Partnership for Tomorrow's Health, which I co-lead with John McLaughlin out of the University of Toronto. And then I'm going to tell you a bit about our assets, what we've collected over the many years that this project has been going on for. And then I'm going to give you a taste of the kinds of projects you can do with this activity with some exemplar projects. And then I'll turn it over to my colleague, Dr. Parveen Bari, who leads the genomics arm, the British Columbia arm of this activity. So let's just see if the slides are moving forward. That's great. So just really briefly, CanPath is Canada's largest population health research platform. You're going to get a sense today of what we've collected both across Canada and in British Columbia. And in principle, it is our real-world data or real-world evidence tool for Canadian health. We've collected data from one in almost every 100 Canadians, and in fact, one in every 50 Canadians within the age range that we've recruited from. And there's been a significant investment in CanPath over the many years. Now, one of the reasons that we build population cohorts, both in Canada and across the world, and you're going to be hearing about other cohorts activities across the world today as well, is that they're effectively the population laboratory, right? We get to understand using these population cohorts why some people develop diseases, why some people develop chronic diseases, and why some people don't. And this is just to give you a real quick snapshot. I'm not going to get into all of these numbers here, but this gives you a sense of the incidence, if you like, of morbidities of say, things like cancers, COPD, or heart disease. And so one of the aims of building CanPath is to collect as much information from our participants at enrollment and then also capture information longitudinally or over time. So in CanPath, we've consented to, actually we're at well over now 330,000 participants, but we've consented these participants to allow us to recontact them over the next, say, 50 years, in fact. So at enrollment, we capture, say, this kind of information that you see in this slide, where we capture, say, medical history, participant demographics, other potential environmental factors like that include, say, sleep or alcohol usage, the kinds of things you wouldn't necessarily capture with, say, with electronic health records. And we think that's kind of critical, because, or very critical, sorry, because the kinds of things that you don't capture with electronic health records are the only way you can capture that kind of information is with population cohorts, and they are the important measures that might lead to having a better understanding of who's going to be developing chronic diseases over time. So really quick snapshot, of the kinds of variables we capture. We've collected over 1,600 variables in CanPath, over 1 billion data elements, if you like. And this is all actually visible on the CanPath portal, and I'll show you how you can get to see the CanPath portal near the end of my presentation. Other information that we capture, say, outside of questionnaire information, is we've enabled some studies like ancillary studies that collect MRI information, and we've captured MRI information from approximately 10,000 people. We're Canada's largest population cohort, and as such, we're integrated with international efforts like the 100,000 plus cohort consortium, which I'm on the steering committee from, and you can see here on this slide some of the members of that cohort consortium. And this is just a quick snapshot of that portal, and you can see here in the 100,000 cohort consortium that we're capturing information of not just CanPath, but say the All of Us program, UK Biobank, and if you're interested in, say, capturing BMI, for example, from people across the world, this is how you might want to get to it as well. So again, just an indication of how big we are. And one last thing I wanted to highlight really quickly was that this thing that I mentioned before, or at the beginning of the presentation, is that we have consent to link out to administrative health records. And that allows us to, say, capture environmental data as well as electronic health record data. And we can do that both provincially and at the national level as well. So one project that I'm going to quickly highlight is going to show how using that information, i.e. that linkages to administrative health data, can enable us to develop new, say, biomarkers in what we call a safe space. In this case would be the population cohort setting, where we can retrospectively go back and ask who's developed a cancer and are there signatures of it that we can see in our plasma, in our data. So one real strong motivation for doing that naturally is that the earlier we can detect a cancer, the more efficacious our treatments are. And while there are current screening protocols for cancers across the provinces and across Canada, we could always do better in terms of that early detection signature. We've been developing liquid biopsies using biologics stored in the CanPath cohorts, and I'm largely going to be focusing on samples from the Ontario Health Study. And this is a quick snapshot, again, of the kinds of information that we can capture when people come into our cohort healthy. We've collected a biospecimen, and by linking to administrative healthcare data, we can then see who came into the cohort healthy and then has gone on to develop a disease like breast cancers or prostate cancers or pancreatic cancers. And so for just the Ontario data, which is all I'm showing, this just gives you an indication of how well we're doing. We've already seen that since the Ontario Health Study has begun, a few hundred, we've actually over 400 people within the Ontario Health Study have developed breast cancers. So they came into the cohort healthy and then have gone on to develop breast cancers. Prostate cancers are around the same ballpark. Pancreatic cancers is a lower rate of incidence. We've only got about 40 individuals who have gone on to develop pancreatic cancers. And to develop liquid biopsies, what we also have in these population cohorts is to be able to match to controls, properly matched with sex and age and other factors as well. And so, just trying to, we've, I think, done a pretty successful job in developing a liquid biopsy that uses cell-free DNA methylation from plasma to identify individuals, and this just gives you an idea of the statistics behind that, of early cancer. And in fact, we can see a signature of breast cancers up to 7 years before those individuals were diagnosed or detected by mammography. In some cases, we were seeing signatures in those plasma samples within months that people went in for a mammogram, but wasn't captured with the mammogram. So it really, again, exemplifies what we can do in these population cohort settings. And we can do this for early signatures of breast. We can do it for metastatic breast. The prostate work we can see here that's doing extremely well, and that's already in print. It was part of a publication in Nature Communications, and we're still working on gathering signatures for pancreatic cancers. And pancreatic cancers, of course, are very important because the earlier we can detect pancreatic cancers, which tends to get diagnosed at stage 3, stage 4, the better. So a good example of how well, this is just a summary of, but a good example of how population cohorts can enable early detection, development, and tools for cancers. On the flip side, we can also use population cohorts to understand who in our population cohorts are developing exceptionally well, or healthy agers. And so we can identify, and this is just a project by one of my PhD students, where we're using single cell omics. And so this is a good example of how the biologics that we have been able to store actually allow for us to interrogate at single cell resolution who might be a healthy blood ager. So we've been looking at some of our participants who are over the age of 80, 90, 100, looking at their blood profiles. We've developed what we call a risk score based on the blood features or phenotypes that we've been able to capture. And so what we've been trying to do there is use those scores to identify in our cohort people who are healthy agers and not so healthy agers, and compare them to people who are younger. Right? And then identify individuals who might be exceptional agers and see if there's something at single cell resolution, as well as genotyping them and sequencing them and looking at their epigenomes, and identify which of those individuals and what the factors are that might contribute to this healthy aging. I won't have time to get into all of this. The data, this paper is in our second round of revision at Nature Genetics, full disclosure. But lots of great insights coming from this particular study. But again, it's just a great example of showing how we've been able to use, say, our blood samples. They've been stored well enough that we can look at things at single cell resolution, and we've been able to develop things like transcriptional risk scores. We've been able to identify sex-specific and cell-type-specific risk scores that might be associated with healthy aging. So I'm going to end off just by thanking all of the funders across Canada. There's funding from across all of our provinces, as well as our national funders as well. I mentioned before, we have a portal. This is our portal. This is how you can get to our portals at portal.canpath.ca. This is how you can see where our data is. And again, I just want to thank the leadership of our organization here. Parveen is on this slide, and he's going to be the next presenter as well. So again, thank you very much.
Thank you so much, Philip, for that great overview of CanPath and some of the great findings that you have identified. As someone who's part of this cohort, I really want to talk to you to see if I'm part of those healthy agers or whether I have a higher risk of cancer. But let's leave that to another time. But let's focus now on Dr. Parveen Bari, and let's hear from him about how the BC Generation Project fits within CanPath. Parveen?
Thank you so much, Susanna. So you just heard from Philip about CanPath and some incredible research being done with this national resource. I wanted to expand on that by focusing in on some of the work that we've been doing with the BC Generations Project, or BCGP, one of CanPath's regional cohorts. So just some background details about BCGP. The study is based out of the BC Cancer Research Institute. We recruited about 30,000 British Columbians between the years 2009 and 2016. Participants were 35 to 69 years of age at time of recruitment. They completed a detailed baseline questionnaire that collected data on a host of demographic, health history, and lifestyle factors. And the vast majority of participants donated blood and urine samples. The blood samples were separated into serum, plasma, red blood cells, and buffy coats, and they're all stored along with urine samples at minus 80 degrees Celsius in our biobank. About 50% of our participants came into assessment centers at baseline where they underwent various physical measurements, including bioelectric impedance analysis to measure adiposity and spirometry to measure lung function. So here's a diagram summarizing the various resources established at baseline. You have the questionnaire data, the biosamples, and the physical measures data. Now, since then, we've expanded the resource quite considerably. We've done a few follow-up questionnaires, getting detailed residential histories and employment histories. We also link annually with the BC Cancer Registry to identify any incident cancer diagnoses among our participants. And as an offshoot of that, we recently established something that we're calling the Virtual Tumor Repository. By allowing us to characterize cancers at the molecular level, tumor samples can support not only detailed clinical research into indicators of prognosis and treatment response, but epidemiologic research into etiology, prevention, and early detection. Now, population-based cohort studies like those in CanPath are an ideal resource for tumor specimens since they're annotated with detailed demographic, lifestyle, and health history data that would support innovative cancer research. So we didn't have the resources to establish a physical tumor repository, but given that pathology departments across the various health authorities in BC, most of which are depicted in this map, are required to store tumor samples for 20 years after pathology reviews are completed, we established a process that can facilitate access to tumor samples that were collected for pathology review from BCGP participants. This involves a number of steps, including review of pathology reports, applications to laboratory services in each of the health authorities, and there are costs associated with these requests as well. But when we tested this out by attempting to retrieve a random set of 200 tumor samples, our retrieval rate was 86%, which is actually higher than many other cohorts around the world that have established tumor tissue resources. So I have a master's student that is using this resource for her thesis. It's a pilot study where she will determine if lifestyle factors captured before cancer diagnosis, things like diet, exercise, smoking, and alcohol consumption, are associated with levels of SPARC, which is a protein whose expression in tumors has been associated with prognosis. The hypothesis being that cases engaged in healthier behaviors before their cancer diagnosis will have tumors that express higher levels of SPARC and thereby have better outcomes. This is a study that could only be done with a resource like CanPath, where you have lifestyle data collected before disease diagnosis. So we brought the tumor samples in, we took cores and created microarrays, and you can see one of those in the picture here. And we've done the staining and imaging and just recently received the data and are in the process of doing the analysis. Now, we've also successfully linked our cohort with other administrative data holdings through PopData BC. We were able to directly link with the Ministry of Health's Chronic Disease Registry, which, for those who are not familiar, uses algorithms combining medical service plan, hospital discharge, and PharmaNet data to identify cases of non-cancer chronic disease in the province. Now, we're particularly excited about this linkage because for studies of incidence of non-cancer chronic disease in cohorts such as ours, we've typically had to rely on self-report through follow-up questionnaires, which is a resource-intensive process that's subject to various potential biases. So far, we've only linked a minimal data set as part of a test run, but are hoping to complete a much more comprehensive linkage of our data in the future. And finally, I wanted to discuss linkage with the Canadian Urban Environmental Health Research Consortium, or CANU, which provides geospatially resolved data quantifying various aspects of the built environment at the residential 6-digit postal code level for our participants. And this is something that actually has been done for each of the regional cohorts across CanPath. So here's an example of a study we just recently published using CANU data. We conducted logistic regression analyses where the outcome was the odds of getting less than 7 hours of sleep per night, which was self-reported in our questionnaire. And we consider this to be an insufficient amount of sleep. And what you're seeing here are odds ratios for each interquartile range increase in exposure along with 95% confidence intervals. And you see evidence that greater levels of green space are associated with lower odds of insufficient sleep, whereas higher levels of light at night, sulfur dioxide air pollution, and living within 100 meters of a main roadway are associated with greater odds of insufficient sleep. And what was really interesting is that the results were largely unchanged when examining each exposure by itself versus considering all of the exposures together in the same model, meaning that each of the factors seems to have an independent impact on sleep. So in the newest iteration of the CANOE data, which we're set to receive very soon, they have included noise data, which is obviously pretty relevant to the question of sleep and is something that we'll certainly be taking a look at. And so where do we go from here? Well, at the CANPATH level, and certainly for us locally at BCGP, one of our major focus areas is to unlock the full potential of the cohorts by unlocking the data that's sitting in our biorepositories. As I've hopefully illustrated, we have high quality data on lifestyle factors, environmental factors, and on health outcomes. But data on host susceptibility factors, for example, remains sitting in our freezers. And so by unlocking genomic data, for instance, we could look at gene-environment interactions with the CANOE data. You could do this CANPATH-wide for truly powerful studies. And this would allow you to identify subpopulations that might be particularly susceptible to the negative impacts of certain exposures, many of which are expected to worsen as we continue to see the impacts of climate change. And so to be able to support these types of precision health initiatives, we really need to generate omics data for our participants. And I just wanted to finish off by acknowledging the BCGP team members who have been instrumental in creating and curating this very unique research resource. Thanks so much for your attention.
Thank you so much, Parveen. Looking at all the different data that you have available within the BC Generations project, as well as what Philip was showing within CANPATH, as a data scientist, I'm itching to put in a data request to have a way to do some analysis in this amazing resource that both of you have put together. So let's now switch on to our panel discussion. It is my pleasure to introduce the rest of our panel now. Tiffany Botwood is the Managing Director of Australian Genomics. She is also a founding director of the Childhood Dementia Initiative. Anastasia Weiss is the Director, Scientific Return to Participants and Impact for the All of Us research program by the National Institutes of Health. All right, so let's begin some of the discussion. Maybe I'll start with Tiffany and Anastasia to ask you about the vision and some of the key lessons, if you could share with us, regarding the Australian Genomics and All of Us, respectively.
All right, would you like me to start off?
Yes, please.
Thank you again for the invitation. It's lovely to be part of this conversation. So Australian Genomics vision has evolved somewhat over the last few years. We originally commenced as a federally funded research program looking at the application of genomics in a diagnostic setting in various rare diseases and cancer projects. And we recruited several thousand participants and in some instance family members into these cohorts and looked at the implications of this genomic intervention for the patient and their families, but also health system impacts. So we were measuring implementation science metrics, we were looking at health economics, really to try and inform health system design. So that was our first 5 years of work that wrapped up in 2020. But as part of that program, we'd established a national research infrastructure for genomics, and the federal government was keen to see that infrastructure leveraged by other genomic research programs. And as such, beyond that first phase of work, we were refunded by the government to provide support of other genomic research projects to improve their efficiency and impact, and also to facilitate translation of research outcomes into healthcare, working with governments to inform policy and practice. So our vision has evolved much more from research into enabling health system, the health system to take advantage of genomic technologies.
Thank you, Tiffany. Anastasia?
Yeah, thanks. And I also would like to thank everyone for the opportunity to talk to you here today about the All of Us Research Program. It's really exciting to see all of the places where we might be able to combine some of this data with all of the work that you're doing in Canada and the places where some of our programs overlap. So the All of Us Research Program is funded by the National Institutes of Health of the United States of America. And our goal is to invite over 1 million individuals who reflect the diversity of the United States to be able to become a part of this research resource that will help with being able to accelerate health research and medical breakthroughs that will be applicable to all of us. We try and do this through 3 main aims. The first is really to think about our participants as partners and nurture that partnership where we are working with our participants and engagement that is a two-way path where they are members of our governing boards, they are a part of our process of developing the information that we share with researchers and what we provide back to our participants, and really engage in the whole process. And we listen to all.
Oh, hey there. I'm Dr. Kaylee Byers. I'm a scientist and a rat detective. For real, I've done some research that quite literally involved following them through alleyways. And now I'm thrilled to say that I'm also the host of Nice Genes, a podcast brought to you by Genome British Columbia. Nice Genes is going to dive into the scintillating stories about genomics. Genomics make up our world. This code of life is quite literally giving me and you life. Your genomic portrait includes all the details in your hair, the food you eat, and even that little feeling deep in the pit of your stomach that only you get when you're about to do something new and exciting, like start a new science podcast. Nice Genes is all about myth-busting and kicking down science-y doors. And while we do that, we'll also explore some of the most exciting topics in modern.
Hello, sorry for that. We are having a bit of a technical difficulty, but we're good to go now. So sorry, Anastasia, you got cut off in the middle of that discussion about the All of Us project. I don't know if you know where you cut off. If you could continue, otherwise we'll just go to the next question.
For me, it was about when you were talking about partnership with patients, with your recruits.
Awesome. So I can just give a brief summary, which is basically that for the All of Us Research Program, we kind of have 3 main goals. One being able to make sure that our participants are partners, that we are engaging with them in everything that we do within the program. That 2, we have a resource that is built for researchers to be able to access the data, and that is broadly available and secure in order to be able to respect the participants' privacy. And then 3, really trying to be able to ensure that all of this information is available for everyone to be able to utilize, and for researchers from the community, students, that all of the researchers reflect the broad diversity of our participants as well. So we want to make sure that overall use of the research is also catalyzed going forward. And that involves working with funders to be able to make sure that data is accessible, as well as ensuring that there are tools and resources available for researchers to be able to utilize the data and make it accessible.
It's quite amazing what has been done in Australia and what is now being done in the US. And so with that, Philip, my question is, given that all of these large cohorts are available, the genomics data is being used worldwide for health research. Some people may ask, why do we need a Canadian cohort? Why are we so special? What would be your answer to that?
Right. I mean, yeah, and you even got a sense of from one of my slides, the International 100,000 Cohort Consortium. There are many large cohorts being built, but one of the things that I would like to emphasize with that slide is that almost every developed and even and some of our lower income country participants are developing these national population cohorts as a way for them to better for those jurisdictions, those countries, those regions to better understand the landscape of health in their individual populations. So yes, while there are great activities going on in Australia and the US and in the UK Biobank is probably one of the most important health programs on the planet, there is only one Canadian instance, right? And there are unique aspects of the Canadian healthcare system that can contribute to health, for example. So it's not just about understanding, for example, the biology of health. It's also about understanding how our population interacts with our specific healthcare system, which is of course unique to our provinces and to Canada. And I would just end off with another statement around what makes Canada unique is we have a distinct geography, just like any other country has a distinct geography and environment. And while we might be capturing, say, similar genetics of certain segments of our population that are similar to that, say, in the US and the UK or in Australia, we also have a unique interaction with our environment. And that unique interaction of our environment, or that genetics by environment interaction, is going to be specific to Canada. It's going to vary across Canada. And it's going to require very large population sample sizes to capture those interactions. Those genetic interactions, or those genetic by environment interactions, have, without getting into too much statistical detail, small effect sizes that can have big impact on morbidities. And lastly, I'll just emphasize too, is that while we do have some commonalities with those other populations with regard to our ethnicities and demographics, we also have uniqueness with regard to our ethnicities across Canada as well. Specifically doing better in the context of some of our underrepresented groups, traditionally underrepresented groups, is someplace that we could focus on further in Canada as well.
Thank you, Philip. Really well said. I think every country has its own, obviously, diverse population, diverse healthcare. We all are different, so it's really good to have a Canada-based cohort where we can hopefully derive some results that are going to be applicable for all of us who live here. Anastasia and Tiffany, we're switching a little bit of a gear. I'm interested in knowing what kind of omics data types or other data have you generated within each of your projects or population-based projects? If you could tell us about it.
Yeah, so I can start us off maybe and tell you just a little bit about some of the data we have for the All of Us research program. We are generating whole genome sequence data, and we currently have over 245,000 whole genome sequences that are available in our researcher workbench. This is combined with other sources of data because, as was just mentioned, it's really important to be able to think about some of the other context related to health. It's not just related to the genomic data itself. So we do also have survey information that's available from over 400,000 participants, information from electronic health records. Our data is obviously a little bit more fragmented in the United States, but we do still have electronic health record.
It looks like Anastasia cut out a little bit. Tiffany, do you mind answering the question about what kind of omics or generally what kind of data you generated?
Poor Anastasia's not having much luck with this session, is she? in the context of Australian genomics, because initially our program was looking at evaluating the impact of genomic interventions in, well, we did 19 different clinical cohort studies across rare disease and cancer. The omics data we collected really depended upon the clinical indication. So, for example, if we're looking at leukemias, we were doing RNA-seq and other various sort of capture panel data and MLPA. Then with, we did whole genomes in some circumstances. So it really depended on the indication. For our large expanded reproductive carrier screening study, Mackenzie's mission, that was recruiting 10,000 couples to look at their whether they had an increased chance of having a child with a severe pediatric disorder. For that study, we used both capture panel or exome sequencing data. So beyond the actual omics data, then we're also looking at, as Anastasia was alluding to, demographic information, ethnicity information, socioeconomic status, education status, and we were very interested in sort of getting a geographic distribution that was representative of our population. Also, we did an extensive psychosocial analysis, so surveys, decision regret for being involved in the studies, really tried to, beyond the sort of omic data, really trying to flesh out participants' perspectives and reactions to being involved.
Thank you, Tiffany. Luckily, Anastasia is back. It looks like the last session is having some major issues with technical about connectivity. Let's switch over to Parveen. It was mentioned the type of omics data that were generated within the other large-scale projects. You mentioned during your presentation that only a selected number of subjects were profiled for omics. How do you think having a fully profiled cohort would help us do better in terms of providing more value for the research community?
Yeah, so I think it's fair to say that there would be really an exponential increase in value. Right now, the way things work is that a researcher with a strong track record of success, comes up with a very specific idea, somehow generates pilot data, applies to funding agencies, seeking large enough pools of money, and maybe after a couple of resubmissions, meaning that it's taken a few years already, they get funding to do an omics study where they end up generating these data on a small number of BCGP participants. Well, if we already had these data generated for the entire cohort, it would really completely shift this dynamic. Suddenly the research enterprise opens up to many more people at different stages of their careers. The availability of this resource would also facilitate better dialogue between the research and healthcare communities. And the research community can also be much more nimble in addressing questions that are of direct relevance to healthcare. And on top of that, you can also start asking highly relevant research questions that might otherwise be very difficult to get funding to address. So with all of that, I truly believe that cohort-wide omics profiling would be completely transformative.
Thank you. So the next question is for both Philip and Parveen. What do you think the other ways are that the research, or what other ways could research and healthcare in Canada and beyond could benefit from CanPath and its sub-cohorts that couldn't be realized yet due to a shortage of resources or other reason? What are the ways that we, the community, could help you achieve those?
I guess I'll start, and then sure. Thanks, Parveen. I think just re-emphasizing Parveen's last point, in fact, with regard to getting some of our material that's been cryopreserved turned into actual data. One of the reasons, it's not the only reason, but one of the reasons UK Biobank is one of the most important health programs on the planet is because it was the first to get genotyping done and genome sequencing done of almost 500,000 people. Now, while that would be a great target for us as well in Canada, that would be important not just from a discovery perspective, but also understanding how our healthcare system can actually utilize or use genome information as well. It's not going to be very long before almost everybody will have a genome available. So understanding in the healthcare system how clinicians can use that information, how a healthcare system can use that information, how we store it, manage it, privacy issues, insurance issues. I think having significant data is going to be critical for being able to maximize how, say, cohorts like CanPath can support the healthcare system. And I'll just kind of end off with one note is that I got to take part in a summit or a workshop in Ottawa a couple of weeks ago. It was called the Environmental Health Summit, and it was fantastic to hear, even from a couple of senators, how they were aware of CanPath's potential for understanding how climate change and the environment can impact the health of Canadians. So that linkage work that Parveen was talking about, I think is going to be critical for that. Those programs like Canoe and how Canoe relates to CanPath and how we can use that to predict, say, where resources may need to be dedicated to support individuals at risk, either it's due to changing water tables or heat, air pollution. I think that's where CanPath has had already significant investment, and we just need to keep continuing to invest in making that data available to the broader research community.
Thank you, Philip. I really like your point about the government is now realizing the amount of value that could be derived from CanPath, and hopefully we could produce additional omics data like Parveen had suggested earlier, so that we could do a lot more with the cohort. Parveen, do you have anything to add to what Philip said?
Yeah, I mean, not much. I mean, it was very nicely said, Philip. And I think that it's the ability to link data, has been pretty amazing. I mean, it's not perfect, as we know, and there's challenges associated with it. But the ability to link administrative data sources, health data sources, to the data that we've collected from the cohort, is already quite a powerful resource. But then to add omics to that as well, and to be able to potentially add clinical data, you really open up the areas of research that these cohorts could really help address, right? It's not just population health research, which is my focus area, but you can actually start asking clinical research questions as well, and that's part of the value we hope we're sort of demonstrating with the tumor tissue bank as well, is that this ends up being a very unique resource to help make discoveries on the clinical sort of treatment prognosis side of things.
Yes. Talking about the clinical prognosis side of things, Anastasia, you lead the responsible return of genomic results to participants. So I was wondering what kind of results are returned to the research participant. Is that something that are actionable for the patients, or rather people who are part of the study?
Yeah, we give our participants choices about what types of DNA results they want to be able to get back. So the participant can decide what's of value to them. Some of the options involve genetic ancestry or traits information, where we provide back information about bitter taste perception, cilantro preference, whether or not an individual is lactose intolerant, and their earwax type. So things that individuals might find interesting that also allow them to engage with genetics and to be able to learn more about how a genetic trait may be hereditary and inherited from others in their family. Then we have information about health-related data that is also available to our participants. And there they have two choices: a hereditary disease risk report, which provides back information based on the American College of Medical Genetics and Genomics secondary finding list. So it's 59 genes that are involved in health conditions and cancers that are potentially medically actionable. And then a medicine in your DNA report, which has 7 genes that are related to pharmacogenetics and how your body might process certain medications. And so each of those are separate choices, and a participant could decide which ones they want to be able to get back.
I love that the participants decide what they want to do, but I also love the fact that it's almost like a bit of a training, so to speak, to the participants to learn about genetics, what the results could mean for them. Tiffany, I'm interested also from your perspective, from your study. Are you returning any results to the participants?
Certainly, and in a similar approach to Anastasia, it is the choice of the participant, and so a sub-study of the acute care genomics study provided participants with the opportunity for additional findings around their own health risk, their child's health risk, and pharmacogenomics. The approach in Australia to additional findings is fairly conservative, so sort of highly penetrant sort of adult onset cancers, Lynch syndrome, breast and ovarian cancer. And, we're not at this stage considering the ACMG list, but, we're really testing the water with participants, and there seems to be, the public is very interested in accessing this information. And there's been high demand for studies in Australia that are providing this sort of future health risk information, which is, it's gratifying from our perspective to see this public interest in receiving the information. I think also to Philip's point about these amazingly rich data sets you have in BC and in Canada, in providing this sort of in the opportunity to understand an individual's health risk and then to extrapolate that to more health system planning, it's, there's an opportunity here for more efficiency in health system delivery as, health systems are trying to cut costs, moving to models more of prediction and prevention. And genomics is absolutely critical to that sort of area around health system efficiency gains.
Thank you. Now I wish that I was also a citizen of the US and then Australia, so I could be participant and ask for my genomic results. So Tiffany and Anastasia, I'm interested to know what are the expected, maybe also some of the unexpected benefits that have been realized so far from the Australian Genomics and from the All of Us projects.
Okay, I'll kick off. Unexpected benefits. So there were certainly unexpected lots of things. The Mackenzie's Mission Study, our large reproductive genetic carrier screening study, we launched recruitment in February 2020, which also coincided with another big global event that we're all aware of and we shan't discuss today. But that forced us to establish processes to enable continued recruitment into the study that were largely virtual. So we established platforms that delivered participants all the education they required, decision aids to help them decide whether to participate or not, and the consent and information materials required for the study and the clinical testing. So those infrastructural elements that we established to deliver a national research program during COVID have now informed the infrastructure we're starting to roll out to deliver healthcare for a large distributed population. And this has really had the benefit of revealing to us opportunities for increased efficiencies in healthcare delivery, genomic healthcare delivery, infrastructure that we will require clinically at scale to deliver genomics at a population scale. but also consumer acceptability and the feasibility of delivering genomics in this fashion. So I think that has been an unexpected insight that we've gained through delivering these studies.
Yeah, that's really great that we could actually push sort of the limits and get genomics more and more into healthcare and patient care. Anastasia, any unexpected or expected benefits so far?
Yeah, so I can say that one of the things that we do within the All of Us research program is for any participant who gets back DNA results, we offer them the opportunity to be able to talk to a genetic counselor. And so this had been intended from the start as being something that would allow for support for our participants and to be able to ensure that they had a resource available to be able to ask questions about next steps and be able to understand all of the material that was being presented to them. And I think one thing that we found, even though it was kind of expected that we were hoping the participants would benefit from this, is that we found that from listening to the participant experiences, they've really talked about how even with the reports themselves having been comprehension tested and providing information in ways that they felt was simple and understandable, that it was really incredibly helpful to have that genetic counseling available to them to be able to talk about how this was uniquely applicable to them and what their next steps could be, and to just be able to ask all the questions that they had after they'd sat and looked through all of the information and the resources that were available for them to be able to freely explore. And then I think something that was a little bit more on the unexpected side is that we did do a number of focus groups and surveys out to participants before we started our DNA results, asking them what kind of information they would most value getting back from the program. And a lot of these surveys really indicated that the participants were the most interested in being able to get back those health-related results. But one of the things that we found so far from our early return is that we are finding that more participants are interested in getting back their genetic ancestry results than anything else. So it is interesting to see that, some of the early information from participants around what they might be most interested in doesn't 100% align with what people do when they actually have the information available in front of them. But we are finding that the participants are overall really highly satisfied with the information they're getting back, regardless of the choices that they're making.
Thank you. It's really good to hear that the participants are so engaged and they give you feedback as to what they found useful and what where potentially they wanted additional information, and I could definitely see the benefit of having genetic counselors explain the results. That would be quite important. So before we go to our Q&A, I want to remind the audience to please ask your question using the Slido panel on the side of your screen. So as the last question, before we get to the audience questions, this is for all of the panelists who want to address it. The learning health system has been a very much talked about subject these days, and it was highlighted as a big need for the Canadian healthcare by the Pan-Canadian Health Data Strategy Expert Advisory Group. In your experience, has genomics played a role or could play a role, and how, in the learning health system? Who wants to start?
I will.
Well, I'll start with Canada. So, from the perspective of CanPath and how it can contribute to our learning health system, I think there's great opportunity and value there. I mean, as I started off, I think in our presentation, it has been, we cannot make a case that CanPath is that real-world evidence opportunity, if you like, for organizations to use the population laboratory to best understand how, say, what factors are contributing to variation in health, how people are responding to our health systems, and so on. We don't have a lot of experience yet, though, with how genomics could support the learning health system in Canada yet. So that it's great to hear what's happening, for example, in the Australian system, and how activities there are being considered at scale. I was just on a call earlier today where it's still considered in Canada that, say, pharmacogenomics could be something of a boutique exercise, when in fact that's quite likely not to be the case. And some of our, if you like, industry partners could, I think, learn from what's happening in places like Australia or the US in terms of how data is being managed in that kind of context and can support learning health. So that's just a start from a Canadian perspective, let's say.
Tiffany, I think you wanted to say something too from the Australian side.
Yeah, well, actually, I'm going to borrow a Canadian perspective too. We had Dean Rieger presenting to us recently and talking about the learning health system model. And I think there is certainly opportunity for genomics to inform health, like intelligent health system design. The problem we all have, of course, is there's sunk costs and existing structures that are not readily suited to taking advantage of advanced therapeutics, of genomic technologies. And we need to develop an understanding within governments about the importance of omics and genomics data in the opportunities around intelligent health system design, the fact that genomics is a disruptive technology in healthcare, in as much as it sort of not only breaks down the traditional systems-specific way of looking at healthcare by organ system. It's pan-disciplinary and it's across the life course. And so we really need a completely novel way of looking at health system delivery in order to generate this learning health system model and really take advantage of the opportunities that genomics can provide.
Thank you, Tiffany. Anastasia, anything to add from your perspective?
I think there's been some great points mentioned already by Philip and Tiffany around the opportunities for being able to have these data sets available. We are hoping for similar things with the All of Us research program, that having this data set available to the community is going to allow for studies that are more reflective of the diversity of the United States, that it allows for being able to combine different types of data that may not have been as readily accessible in the past, as well as for being able to link in other data sets in the future, and that this will help with driving improvements overall in the health system in the future.
Thank you. I guess we're maybe now, unless Parveen has something to add, then we'll switch to the Q&A. No? All right. So now it's the audience's time to ask the questions. So the first one that I see here: are there any specific example of changes to policy and or practice that resulted from the work done by Australian Genomics and All of Us? I think there was some mention of this already, but I don't know if you want to elaborate, Tiffany or Anastasia, on this.
I'm happy to start. Look, we have seen significant changes in Australian policy and practice over the last sort of 7 years as a result of not only the work of Australian Genomics, but other state-based genomic research programs, and also very engaged professional bodies, you know, the genetics colleges, etc., and pathology colleges. Some of these include the publicly funded genomic testing through our Medicare Services Advisory Committee deliberations. So our Medicare now offers a number of genomic panels that is subsidised. We've seen the establishment of a genomic and health technology assessment policy branch within our federal government, which is directly charged with considering the implementation of genomics nationally for Australia. And there's also committees that have been established between our jurisdictional departments of health and our federal Department of Health that are exploring opportunities for implementation of genomics locally within the jurisdictions as well as federally. So there's been a great appetite in our governments to consider how genomics can enable better health delivery for Australia.
Yeah, that is really amazing to hear that these larger studies are leading to actual changes. That's what we hope for, right? But you never know if it's going to happen. So it's nice to see when it actually does. Anastasia?
Yeah, so I can say that Tiffany's story is definitely a great example of how powerful these types of cohorts can be. I would say from the all of us perspective, we haven't seen that level of change around federal policy, but we are seeing that we're starting to have individual healthcare providers and others starting to cover more of some of the genetic testing and some shifts in some of the receptiveness to being able to include genomic information in care throughout the country.
Thank you. So we'll go to the next. I think it's not quite a question, but I think it's more of a statement that the lifestyle data collected before disease diagnosis seems like something primary care physicians would benefit regarding family history. As it was mentioned in the previous session, some people, new immigrants or those who maybe parents have are not known or potentially deceased, it's hard to know family history. So hopefully, I don't know how, maybe you could comment on how the lifestyle data could contribute in your studies or did contribute to some of the analysis that were done when family history was missing.
Well, I was going to say something not so much about family history per se, but other lifestyle factors that were asked about. And one of the things that became interesting for us was at CanPath, as many people may have heard, and I even saw this in the New York Times, is that there's been an advisory that with respect to alcohol usage, that people limit the number of drinks they have per week to 2 drinks a week because of its risk for cancer. And so that's a kind of information we can naturally capture. It's been long known that alcohol is a cancer risk factor. So it's one of those things where we've been able to explore in CanPath how that contributes to cancer risk. And naturally, we do see that relationship. In fact, that 2 drinks is right at that threshold by which risk for cancer increases significantly. And further, we've been able to look for that in the context of how can that support decision making with regard to early detection and prevention. So being a bit more aggressive, if you like, for monitoring monitoring disease. Family history should naturally be part of particularly for cancers, should naturally be a question asked by a physician with regard to being an important character trait with regard to risk. And so I think that is often something that's routinely done. So,
Anybody else would like to address that?
I think not the Australian example, but a lot of the Nordic nations where health data is not only collected throughout one's life course but accessible to an individual. It's a demonstration of not only empowering sort of policymakers and health decision makers and health resourcing, but also empowering the individual in being able to consider, if they had a pediatric cancer, for example, how do they manage throughout their life, or look back at what specialists they saw at different stages. I think, the longitudinal data collection, health data collection, and then opportunities to link that to other data sets, as we've heard today, that would be such an aspirational goal for so many nations in terms of empowering the system, but also empowering the individual.
Well said, Tiffany. If there is nobody else who wants to take that question, we could switch to our next one. Can rare genetic disease patients could benefit from your work? I think it's a really good question. Anybody wants to take that?
I'm not going to go
I can
first.
start off.
All right.
So
Anastasia, maybe.
Yeah, I'm happy to start us off. So
Okay.
within the All of Us research program, we do have, some cohorts of rare disease patients that have joined the program and some conversations that we've had with those communities. While the results that we're returning back are not necessarily specific to any rare disease condition for all of the many different rare diseases that are out there, having the information available to researchers and as part of that whole genome sequencing data combined with the electronic health records data that provides information about their condition and all the tests that they've received, the information that they're able to provide throughout the surveys that they complete, and then to make that available to researchers, we're hoping will allow for additional research on some of these rare conditions, even though we're not specifically sampling for any particular condition within the All of Us research program.
I think that's a great point that Anastasia just made. I'll just add to that. I think what we've heard from decision makers is that, and this exemplifies the need for having from a research perspective, having a large cohorts, is the activity you may be doing. Even I use pancreatic cancers as a example earlier, and I'll come back to that again because it is a relatively rare cancer. But the kind of data we can capture only, say, in Ontario and or Quebec or any one of our provinces is still going to be fairly small. It's only by combining information across Canada or using large population cohorts or combining our data with other activities around the world are we going to have significant numbers to do the science that we need to do with those rare conditions so that we can better understand how the factors at play, whether it's a genetic factor or whether it's an environmental factor. So I think it just speaks to the fact that the population cohorts can help support rare diseases. And then the last thing I might want to say about that too is that if there's a disease of where the etiology is complicated, these population studies can potentially identify common factors that can help identify potential treatments that are maybe not directly associated with that rare disease or that disease if it's the etiology is complicated, but there may be a repurposing of treatment that can come from having these large population studies. And again, you need big data to be able to support that using either molecular tools or computational AI tools, for example.
Philip, I really love that point because usually, most of the studies focus on a specific disease or a handful of things. And so these incidental findings, you probably, you wouldn't find when you're just focusing on a smaller set of things. But in a population-based study, you may be able to help one disease based on what was found in some other disease conditions. So that's a really good point. So going on to our next question from the audience, what strategies have you employed to ensure participation by underrepresented groups? Do you track diversity data, and what kinds of changes are you seeing over time?
Who would like to take that one? Yeah.
Yes, please.
So we are implementing, so Australia has a very diverse population. We're largely a migrant population. Our First Nations peoples, the Aboriginal and Torres Strait Islander peoples, represent nationally about 3% of our population. And I suppose there's been a number of strategies to try and improve the health inequity for underrepresented groups in Australia, particularly over the last 10 years. So in the context of our Indigenous peoples, there's been an increasing understanding that the sort of, colonial approach of offering genomics to these communities is inadequate. So we're moving to models where genomic clinicians and researchers and experts that are of Aboriginal and Torres Strait Islander origin, they are leading the work, they are co-designing materials for communities and prioritising research based on community wishes. So that's one aspect. A second aspect is ensuring that we develop genomic resources, information materials, consent forms in languages and culturally appropriate, not just direct translation, but actually working with communities to develop resources that address their information needs. And also targeting recruitment to underrepresented communities to ensure inclusion in research, because the ongoing issue we have is that those underrepresented groups, unless we effectively and proactively reach out to them, they will remain silent and unaddressed, and then their needs will not inform health system design.
Yes, Tiffany, perfectly linked back to our first session of the day. And it's really good that you mentioned the consent. It's not just the language that's being translated, but actually the context, and it's culturally suitable. Because I've seen a lot of research studies, at least here in Canada, the patient consent is done in English, but as we discussed through the session, there is a large immigrant population here and elsewhere too, and so language barriers tend to be an issue. And with that, we're excluding some of the population. So it's really nice to hear that there is actually steps made to translate the consent. Anybody else wants to talk about?
I can speak
Yes, thank you.
a little bit to that one as well. So within all of us, I can talk a little bit about what we do on the participant side and then on the researcher side. So on the participant side, we really work with being able to engage our participants and have a group that reviews all of the materials for our DNA results that we're providing back out to participants, that's made up of representatives from all of our different community partners that reflect the diversity of the cohort. And they look through all of the materials, all of the visuals that we're using, and really try to be able to determine, is this going to be both respectful of the community? Are there places that we can change the language in order to be able to feel more inclusive of everyone? And are there places where we can provide a little bit more information or context? And I can give just a simple example of something there where when we were looking at some of our information we were providing back around bitter taste, our cultural awareness committee really looked at those materials and were like, well, the foods that you're using, we want to make sure that they're going to be examples that anyone could relate to and understand as being a bitter food. So we want to make sure that you're giving lots of examples and that they're going to be reflective of all of those different cultures and communities. And then on the researcher side, we do a lot of outreach into the researcher community and are actually tracking the diversity of our researchers as well, because we want them to be as diverse as the participants that are a part of the cohort. And so there we do a lot of communications to be able to do trainings. We have office hours that are available to be able to learn more about how to be able to access the data and then take that information in order to be able to learn more about how can we make this data more accessible to community researchers or to students who might want to be able to access the data, and what kinds of tools do we need to be able to develop? And that's led us to consider, for example, different types of programming languages that we might make available as tools in the researcher workbench.
Yeah, really good additional points there around not just the research participation, but then the results when they're returned, that they're culturally safe and even with the taste, being able to link it with foods is quite nice, as well as the EDI of the researchers. As session one highlighted, it's very important that we have researchers doing the research on participants where they feel that they're part of the community. The community-led research is quite an important aspect. So it's really amazing to hear that all of that is taken into account. Anything on the CanPath side that may be different?
Yeah, I'll just really quickly give some insights, and what I will say that to some extent too, we've tried a number of different ways to do directed, targeted recruitments, and largely in partnership with organizations or scientists who already have community engagement or community level engagement with some of the communities that we're interested in trying to improve our, if you like, our representation. The imaging project that I just mentioned before included a significant number of self-declared Indigenous participants, and again, that was done in partnership with organizations who already have, if you like, significant community engagement and have had those trusted relationships for many years. So, one of the things that we are also conscious of is that it's something of a learning process too, because we're looking at what works and what doesn't work, in fact, in terms of how we engage with our participants. And one of the things that we've already tried to emphasize, and I think you just touched on this a moment ago, is that those engagements usually need to have representation from the research side of people who are from those communities as well.
Perfect, Philip. Also thinking of, the BC Generations project that's part of CanPath. Just remember seeing the map of BC and was very excited to see that the northern communities, the more remote communities, were also involved. So I don't know, Parveen, if you want to comment around that.
Yeah, so all of the recruitment activities for BCGP happened well before I arrived, but there was a huge effort undertaken to ensure that we got broad representation across British Columbia, and I think for the most part that has happened. The demographics of our study population are not a perfect mirror of the demographics of the province. There's, I think, with any of these studies from around the world, there's challenges in trying to ensure that these studies are representative. And I think one of the things that we've discussed at CanPath as well is whether there would be opportunities to really support some much more innovative approaches to ensuring that we can to enhance representation of these communities, typically underrepresented communities, in these cohorts and in the future. And it's important to make sure that recruitment materials and consent forms, these things are culturally sensitive and they're in languages that are accessible. But to even look at structural barriers to participating in these studies, and I think there's a lot of pretty amazing research that's been looking at how to enhance participation, and I think there might be some opportunities to really employ that to expand representation from these various groups.
Thank you, Parveen. So we're getting close to the end of the session, so I'll ask one last question from the audience. What safeguards are in place to prevent the misuse of genomic data by bad actors or even insurance companies? So I'm guessing it has to do with data governance, but if any of you want to address it.
I'll start with the Canadian perspective, and one of the things that we've had now, it's been on the is now law, is an Anti-Genetic Discrimination Act that came into being, I think it was in 2018 or 2019. So that's something that's quite, I think, important for Canadians to recognize, is that there are measures now in place that are there to protect genetic information so that it doesn't get used by, say, for example, insurance companies to be prejudicial. With regard to genetic data, like all healthcare data, naturally in CanPath we have our data protected behind firewalls, and that are there to ensure that data isn't shared in a way that's not meant for any alternative use by which we allow it to be used for research purposes. And so we do have mechanisms by which we allowed in certain individuals who are bona fide researchers to have access to that data. So.
Yeah, that's really good to hear that there are safeguards in place. We can sleep with no issues around worrying how our genomic data may be compromised. So this concludes our last session. Thank you so much, Anastasia, Parveen, Tiffany, and Philip, for your participation. I know some of you have called in really early in the morning. Some of you are later at night. I really appreciate your time and input. It was just amazing to hear what has happened in the last, decade or so. When I started in my journey of a translational researcher 20 years ago, I could not have imagined all the amazing things that, happened in terms of the amount of data available, amount of people that are participating and just loving the participation, like actively being part of these studies. It's been a really great learning experience, and it was also a great overview of the benefits of the population-based genomics cohorts that could have on research, but also on patient care. I hope we have highlighted our own Canadian cohorts, such as CanPath, and its sub-cohorts could reach the same potential by having additional support from the community. We have 1/3 Canadians as part of CanPath with samples available for omics profiling. I hope in the next few months you can join forces with us to help them achieve this amazing resources potential. Now I will hand it over to Suzanne Gill, who is our President and CEO, for some closing remarks.